Welcome, everybody. Thanks for joining us this morning. This is the Distinguished Seminar Series, which features presentations by outstanding thinkers and scientists that's sponsored by the Allen Institute for Brain Science. The distinguished speakers are selected based on their impact in interdisciplinary research into the neuroscience community. Today, we're really excited to host Dr. Aman Salim. Aman is the Sir Henry Dale Fellow and Associate Professor at the University College London. He did his PhD at Imperial College London with Simon Schultz, studying information process in visual system, working in both blowflies and mouse. This is how you know he's extra cool. Then he went on to do his postdoc with the Harris and Carandini group, studying the integration of visual motion and locomotion in the visual cortex. And he published a terrific paper back in 2013 showing that neurons in the visual cortex are tuned to the running speed of mouse that I think was really impactful in the field and on some of the work that we've done. Today, Dr. Salim will present Vision in Action, Visual Processing in Active Behaviors. So welcome, Aman.
Thank you very much, Saskia. And I'm gonna start sharing my screen. Yeah. Well, first of all, thank you for the invitation. It's a real honor and a pleasure to be presenting at the Allen. It's a shame I couldn't do it in person, but these are the times. So today I'm going to be talking about vision in action. And I'd like to start off with this quote from a very famous pioneering vision scientist, J.J. Gibson, which says, "The simple assumption that perception of the world is caused by stimuli in the world will not do. We look around, walk up to something interesting, and move around it so as to see it from all sides, and go from one vista to another. That is natural vision." So we have known for a while now, so we've known for a while that much of our visual experience is actually generated by our interactions with the environment. Yet most of the research, vision research, until recently has been done in something like this, where you've got stationary subjects. And the reason for this is not, is because, well, you can get very precise control over the stimuli that the subject is viewing, and so and also know what exactly is where exactly they're looking. And also, given that they're stationary, there's fewer behavioral states that you have to work with, and so it makes the analysis a little bit easier. However, now with all the advances we've made, we're in a much better position, and so we can now look at vision in action. And so today I'm going to be talking about 4 different topics from the lab. The first one is about a new software we've been developing to study vision in more interesting scenarios. I'll talk about some visual coding during navigation, and then responses of the visual cortex to sudden perturbations. And finally, I'm going to be talking about an anatomy study that we ran in the lab. So first, to start off with this new software, I want to start off with the background as to why we started this whole thing. There's it's not something where who wants to build a better mousetrap. There's a lot of softwares already out there. Why did we decide to do this? So first of all, when you're studying vision, there's a lot of different stimuli we might want to present. So for example, there's classic visual stimuli that have been used for decades, where you want to characterize things like tuning curves, which are usually defined as pixels on the screen. There's also virtual reality environments, which are being increasingly used right now, which is based on rendering different 3D worlds. And finally, there's more complex environments, which we might be starting to use soon, which are something like augmented reality systems and so on. So when I started my lab around 4 years ago, I faced the same question that probably most vision scientists usually face, which is, which software should I be using to generate visual stimuli? Ideally, we want the software to be a single software which can run all these different types of stimuli so we can switch between them. We want it to be accessible and easy to use, so it's either new members of the lab can take it on quite easily, or we're able to share our stimuli quite easily to the rest of the world. And finally, because we're doing a lot of instrumentation and trying to do various recordings, it's good to have it in a way that's very easy to integrate with this instrumentation. So at the same time, while we were playing around with different instrumentation, we started exploring various open source tools for the lab, and we started exploring Bonsai. And that was really useful in terms of getting a lot of things going quite quickly in terms of like data acquisition and so on. So we contacted Gonzalo Lopez, who is the developer of Bonsai, and explored our options in terms of can we actually use Bonsai to generate visual stimuli. So that's where the journey started. And the first task was to try and come up with a framework where we can have the 3D stimuli and 2D stimuli, the classic visual stimuli, within the same framework. So first, in terms, so classic stimuli are usually defined in terms of elevation and azimuth, so in terms of visual angle. So how do we get this in? So we decided to try and take this onto a 3D environment. And we did this by taking each of these positions and projecting them onto a unit sphere. So so a stimulus like this gets wrapped around the sphere in the way that's shown out here. So this uses Mercator projection framework, which is the projection framework used to generate maps from the globe. So once we have this, we can now use it. Visual displays become windows into this unit sphere. So if we, this is an example of 6 different windows. So for example, if you have the display positions here and here, you can see that the corresponding changes in the visual display. And as you move them around, the stimulus adjusts according to the requirements there. Sorry, 1 minute. So we can use this to then actually try it out on actual in real environments or experimental environments. This is an example of the stimuli. So in this case, we've got the checkerboard stimulus. And for the 3D stimuli, we've got different objects placed in a 3D world. So you can see that the stimuli are wrapped around on the screens appropriately. If we go to, we can also try a different display. In this case, we tried it on a head-mounted display for human subjects. And again, you can see that there's more, because these screens are closer to the eye, there's more warping of the stimuli here. And finally, we also did it on a dome, projected onto the inside of a dome. So you can see that in this display. And all of these things have basically the same underlying structure in terms of generating the stimulus. All that's changing is the actual display end of it. So given that actually to be able to define a stimulus, what we need is the position of the screen, we actually decided to try and automate this. So here is an example of placing a marker. So we just take a printout of a marker and place it in the position where maybe an observer would be sitting, and then presented a marker on the screen. And we just take an image of this. And Bonsai then calculates the 3D position of the screen with respect to the observer. And you can get an appropriately warped image. If you move the screen and then retake a new image, you can get an appropriate warping as well. So this was, we're quite excited about this because it avoids us painstakingly calculating all the distances and angles of the screen in usually quite inconvenient experimental rigs. And instead, we can just take a picture using a camera. These were actually taken using a mobile phone camera. And then you can actually calibrate your stimuli. So does this all work? Here are some example experiments. So this is a case where we presented a very simple behavior. This is a small dot moving overhead above the animal. And this elicits a freeze response, as we've shown before. So we've basically replicated some previous results in terms of generating animal behavior using this, using BonVision. We've also characterized whether we are able to do electrophysiology. So this is an example of getting receptor field maps using sparse noise stimuli. And as you know, sparse noise is quite sensitive to timing issues. So it's effective enough to recover good receptor field maps. We also can generate virtual reality environments quite easily. And this is an example of a very simple virtual reality. It's a virtual platform that the mouse runs on. And these lines here represent the speed of the animal across the platform. So on the first 2-3 days, the animal is basically increasing its speed gradually as it's getting more familiar with the environment. But interestingly, by around day 5, they start developing a certain speed profile. So as they reach the end of the core platform, they tend to slow down innately. So there's no reward or anything here. So that was quite interesting. So we're able to generate stimuli that are immersive enough for the animal to feel like it's approaching towards the end of something. We also did some human psychophysics to see if we are able to do some depth perception effectively. And this was using a head-mounted display and trying to distinguish the size of the cubes, and human subjects are able to do this. So these are all pretty standard experiments that have been run, but what's interesting is that we can now run them within the same framework. So virtual reality and the classic stimuli can be run within the same framework. But also we can extend it to other things. Now, given that the observer and the screen position and the observer position can be altered, we can actually do that online. And this is an example where the image on the screen is updated based on the position of the subject. Well, why is this interesting? Well, we can now create things like this, where imagine this is an environment where there's a tree outside the room and this is a window. So if a subject is in this position of the room, the window would have the tree appear around this size. But if the subject then moves to the end of the room, the tree would actually appear bigger. So you can try this out if you have a window close to you now. But basically, the image on the screen, the scene on the screen needs to, depends on the viewpoint of the animal or the animal position. So now we can actually take advantage of other tools that are developed in Bonsai. And in this case, we are using DeepLab-Cut. We can run inference models of DeepLab-Cut trained networks to try and get an accurate position of the animal, and then update the scene on the screen. So I'm going to show an example of this in a movie. So this is the animal moving around this environment, and we are tracking the position of the head of this animal in the environment, and that's shown in red here. And you can see as the animal is moving around, we're able to update the scene in real time on these screens. And so now we can actually generate these kind of augmented reality environments. And it's actually quite easy once given the framework we have, because this is all about just putting the screens in different positions. And then the other thing is we can actually import scenes that are quite complex environments that are built in Blender, which is an open source tool for creating 3D environments. So just to summarize this bit, so we developed this new software, which is an open source visual environment generator called Bonsai. We're able to generate 2D and 3D stimuli with almost equal ease. And we can get precise control over the stimuli even when the observer is moving around, which allows us to do augmented reality, virtual reality, or classic 2D stimuli. One of the key things that we realize it also with the kind of calibration that we have in place and the way we define stimuli, it's actually very easy to replicate experiments across different labs or across different rigs within the lab. So as I showed you 3 different rigs, so the head-mounted display, the 2 screens, and the dome, we're actually using the same code. We're just changing the end element of it. And it's also naturally closed loop, given that Bonsai, it's based on Bonsai, and that has a lot of easy interfacing with many tools in neuroscience right now. So I'm now going to move on to the actual science, which is I'm going to talk about visual coding during navigation. Now, why is navigation interesting? Well, we spend a lot of our time, at least in regular times, moving around from one place to another. And when we do that, we often use vision to guide our movements through the world. So we're constantly using vision for navigation. And also rodents are known to be using vision for navigation. So place cells are known to differ based on how we change the visual scene. So how is vision during navigation? So we can ask this question in a slightly different way. Does visual processing change in any way depending on the spatial context? And again, we can ask this as in, do identical visual stimuli presented to the animal elicit the same responses when they're presented in different locations? Now, this is really easy to ask in VR. So we move to VR here, and we created a virtual reality which has two landmarks placed in two different positions of the room so that we create two identical sections of the room. So if we do this, move the environment by 40 centimeters either way, we get a repetition of the stimulus. So if we look at screenshots that are 40 centimeters apart, you can see that the matched screenshots look similar. And if you generate a very simple model of a complex cell of a mouse and run that model through the environment, this is the kind of response you might expect, where you've got two equal bumps in the response, which correspond to the separation between the two identical segments of the environment. So what does this look like in the cortex? This is how the environment looks in reality. So this is the mouse running through the environment. In this case, it was a dome. And you can see mice actually love running through these environments. They're quite happy to run often. So we did 2-photon imaging recordings for these experiments, and we focused our regions of interest over the primary visual cortex. This is an example of a single neuron as the animal runs through the environment across different trials. So each line here is one run through the environment. You can see that the responses are quite reliable in this neuron. And also, there seem to be two peaks that are around the right distance away, so around 40 centimeters away from each other, suggesting that the repetition is causing a repetition of the responses. So what do other neurons look like? So the top two neurons here look like what we might expect, that they have a repetition of the responses across the identical visual landmarks. But then we also have these other neurons where there's almost no response in one of the positions, even though there's an identical visual stimulus in that other position. So these are just some very select examples. How does it look across the population? So these are all the neurons that we recorded from, lined up based on their central peak as calculated in the odd trials. And now we look at the even trial responses. And this is the response. So the red and blue lines here indicate the identical 40 centimeters away from the peak. And you can see that at these positions, the responses, the fluorescence is not as strong as in the central peak. And if we quantify this, and here we're looking at the spatial modulation ratio between the two peaks, we find that V1, so the dotted line here represents that a response where the two peaks are equal. And we find that the median of V1 responses around here, it's much lower than this expected response based on visually identical, if they were responding in the same way to the two landmarks. So in general, we have this question, does visual processing depend on spatial context? And the answer seems to be yes. Identical visual stimuli in different spatial locations elicit a different response in V1. So there seems to be some spatial modulation based on position. Our next question was like, is this related to the spatial signals that we've known for years now that exist in the hippocampus? So to get at that, we moved to electrophysiology, and we did simultaneous recordings from the primary visual cortex and CA1 region of the hippocampus. So here's an image of the recording sites in one of the animals. This is the responses in these two areas, so in V1 and CA1, and you can see the responses tile across the whole corridor in both these regions. And given that we have these large populations of neurons, we can then use it to try and decode the position represented by these neurons. So this is red here represents a very high likelihood of decoding a particular position, and blue a very high likelihood of not decoding a position. So you can see that most of the, it's red along the diagonal, which is a dotted black line, suggesting that indeed we are able to decode quite well in both these areas the position of the animal in the environment. But the question, the reason why we started these recordings was, are the two areas representing the same information? And to get at that, we looked at, are they actually making the same mistakes? So there's no reason why these two areas should be making the same mistakes unless they're encoding this similar information. So we looked at that, and on the x-axis here is the correlation, the error correlation between these two areas that we measured. And you can see that there's significant non-zero values in this error correlations. And when we compared it to just shuffling the responses based on common speed bins, we see that we lose these correlation values. So it's not just a result of correlation due to speed. And overall, there seems to be a significant correlation between these two areas, suggesting that they're actually representing similar information. So is this spatial signal related to the hippocampus? Signals in the hippocampus? And it seems the answer again seems to be yes. The spatial signals in V1 are correlated with those that are present in the CA1 region of the hippocampus. So I've so far been talking about spatial modulation in the primary visual cortex and the hippocampus. But we know, for example, that information is reaching the visual cortex from the eye going through the LGN, which is the primary pathway for visual information entering V1. And also, there are actually no direct connections between the primary visual cortex and hippocampus. So there's some kind of intermediate areas that are transmitting information between these two areas. So this was a project led by Mika Diamanti, who then tried to look at the spatial modulation across areas, through areas across the hierarchy of this circuit. So the first thing we did was, for the LGN, we injected GCAMP into the LGN and recorded boutons that express this GCAMP in the layer 4 of primary visual cortex. And for intermediate areas, we focus on higher visual areas. And for that, so this is an image of the boutons. You can see that there is a higher density in layer 4 of V1. And so that was the region that we were recording from, activity from. And for higher visual areas, we just focused our regions of interest over various higher visual areas around V1. So what does the spatial modulation, what does it look like? So here's 3 example neurons from LGN. And you can see that actually it was very rare for us to find, actually we never found any cells that had like a single peak in their responses. There was often multiple peaks. And these are 3 representative examples from the 3 percentile ranges. And if you look at this, the population of all the boutons here, you can see that the side bands are as dark as the central band, suggesting that the second peak is almost as big as the central peak. And if you look at the spatial modulation index, where 0 means that there's no spatial modulation and a high value means that there is a spatial modulation, we find that V1, as we showed before, has a high spatial modulation, but LGN has very little, close to 0 spatial modulation in our recordings. So these are LGN boutons. What about higher visual areas? When we look at them, they actually look very similar to what we observed in V1. So we've got neurons that have dual peaks, but we also have neurons that have just a single peak or a very small second peak. And you can see here, it's quite nice contrast with the figure above, where you see that the side peaks are much dimmer compared to the LGN responses and compared to the central peak. And if you look at the spatial modulation index across the population, you find that they're very close to what the V1 spatial modulation looked like and much higher than what we observed in the LGN. So overall, it seems like spatial modulation seems to start in V1 and then carries through different higher visual areas. I mentioned different higher visual areas. Out here, I've just clumped through all of the higher visual areas. If we actually split them out, this is what each of these areas looks like. So you can see that most of these areas have distributions that are quite close to V1. All of them, none of them was significantly different. And if we look at the distribution on the right here, almost all of them have high non-zero values, barring LGN, which is very close to 0. So overall, it seems like spatial modulation arises in V1 and persists through the different higher visual areas. Now, one thing we were really curious about is that I've been talking about spatial modulation during navigation, but is navigation actually required? Because the animal could just be running through the environment. And so that's something we tested next. So is active navigation actually required to see this spatial modulation? So what I mean by that is, like, when an animal moves through an environment, there are movements of the environment that you might expect based on your own movements. And this is something where as I move around in different locations, there's optic flow and there are landmarks moving in accordance to my movements. And this I would call something that's controlled by the animal and what we would refer to as active navigation. On the other hand, it's also possible that there are movements of objects and things around me that are out of my control. And that would be something which is controlled externally. It's not linked to my own movements. And that's something you would experience if we just replayed the movie of what the animal previously did back to it at a later point in time. So this is what I'm calling replay. And in the next slide, I'm just going to be showing you what happens when only time points when the animal was actually running as opposed to stationary. So it is replay when the animal was running. So this is what it looks like. On the x-axis here is the spatial modulation index when the animal is, when we replay the stimulus to the animal while it's running. And on the y-axis here is a spatial modulation index during virtual reality or when the animal is actively navigating. And what you can see is that the spatial modulation in both V1 and in higher visual areas tends to be about the diagonal. So active navigation seems to enhance the amount of spatial modulation present. And it's particularly strong in higher visual areas. So overall, it looks like active navigation is enhancing the amount of spatial modulation that's present in these different areas. Okay, so to summarize this part, I've shown you that spatial context seems to modulate the activity in the visual cortex. These spatial signals are correlated with what we observed in terms of spatial representation in the CA1 region of the hippocampus. And spatial modulation arises in cortex and seems to persist through the various higher visual areas. It's not present in the LGN, but starts off in cortex. And finally, this spatial modulation is strongest when the animal is actively navigating the environment, as opposed to just running and experiencing the flow of the environment across its eyes. Okay, I'm now going to move on to the third part, which is about responses of the visual cortex to sudden perturbations. Now, as I mentioned before, when an animal moves through an environment, you can expect a certain correspondence of objects moving around you and also correspondence of optic flow around you. So there is a certain expectation that you can create regarding the optic flow that you experience. So a few years ago, Georg Keller and colleagues ran this interesting experiment where they had the visual flow follow the running speed of the animal. So as you see here, the running speed and the visual speed tend to follow each other. But every now and then, they would have a sudden stop of the visual stimulus. So there was a sudden, for 1 second, they would suddenly stop the flow, and then it would go back to being in closed loop with the animal's actions. And when they looked at V1 activity during this period, they found this interesting result where there was a very strong response to the sudden stop in the visual flow when the animal was running. But instead, the green line here is the playback condition where when the animal was stationary, the responses were much weaker. What they attributed this was to sensory motor mismatch or predictive coding, which is the, in a sense, the calculation of what is the efference information coming from the motor system. You use that and then you compare it to the sensory system and you look at the difference between these two to try and predict what is the response to then be able to control the future actions. Like we asked before, we were curious about one thing, which is like, is the active aspect of this necessary? So does the closed loop action stimulation, is it actually required to be able to see these responses? So to ask that, so another way of asking that is like, can purely visual perturbations generate such strong responses in the primary visual cortex as was reported? So this was the motivation for Tommaso Muzzu, who's a postdoc in my lab. And he generated this stimulus which replicates the visual aspects of this sudden stop, which is you've got a grating that's drifting, and then every now and then it suddenly stops for a second. Very similar to the other case, except this is just running independent of the animal's actions. Now, rather than having the animal see a lot of starts and stops of the stimulus, what we did on each trial is that we ramped up the contrast of the stimulus and then ramped it down. So then the animal doesn't see a sudden start and stop of the drift, but instead just sees a constant drift, which is the contrast changing. And during the high contrast periods on some trials, about 25% of the trials, we randomly introduce these sudden perturbations. We did this by presenting the stimulus on one hemisphere of the animal on a hemispherical dome using BonVision. And by the way, full disclaimer, for the virtual reality, those experiments started well before we started BonVision, the development of BonVision. So they were run using PsycToolbox in a custom written program that I'd done over my postdoc. But anyway, back to this. So while the animal was running through this while the animal was experiencing this stimulus, we recorded using extracellular electrodes in the primary visual cortex of the mouse. So what do responses look like? So in this, each row is the response of is a normalized response of a single neuron. And this is all the units we recorded from the primary visual cortex. And red here represents a very high response and blue represents a low response. And you can see right from the outset that there is a very strong response to the sudden stop or the sudden perturbation that the animal is experiencing. If you look at individual neurons, the gray lines here are the responses without perturbation, and the red lines here are responses with the perturbation. So you can see that there's a very strong responses in these animals, in these neurons. We also have neurons where there's no response, and finally neurons where there's a decrease in the response as well. So if we quantify this, and the way we quantify this is, if you take a neuron's activity, can you tell based on a classifier whether or not it was a perturbation trial? And we did that, we got about 60% of the units would be able to differentiate with over 95% accuracy that they were whether or not it was a perturbation trial. So they were encoding this perturbation period. And out of these neurons, more than half of them were positively modulated. So responses like these two neurons here. So there's quite a significant portion of neurons that are actually active to just these visual perturbations. So when we go back to this thing, can pure visual perturbation generate strong responses in V1? The answer seems to be yes. You can actually get a very strong response. But one of the other features of sensory motor mismatch was that it was modulated by running. So we next asked, is this perturbation response modulated by running? So we just split the trials based on whether or not the animal was running during the different periods. And this line here represents the stationary responses, and this line is the running responses. You can see right from the outset, they're very offset. So there is a strong difference between these two. And moreover, if you actually look at the modulation, which is the difference, the degree to which the responses are changing between these two, this is the modulation index calculated for stationary on stationary trials, and this is the modulation index calculated for running trials. And you can see that a lot of neurons have significantly larger responses when the animal was running as opposed to when stationary, and this was quite significant. So overall, there seems to be a high modulation of these responses by running activity. So, well, this still doesn't say that this is not sensory motor mismatch, because maybe the animal thinks that the stimulus follows its own movements, because when it's running, it might be it might happen to correspond to its own movements. So it might, and there's years of sensory experience where the animal's movements kind of follow its own actions. If this was the case, what we might expect is the responses should be highest in the direction that corresponds to the running. And in this case, this is the nasotemporal direction. So if you're moving in and out, that's going to be in this direction. So as the animals are moving forward, you expect optic flow to go from the in the nasotemporal direction. So this is what we looked at. We looked at the average response across all perturbation units. And when we look at that, this is the response. So red here represents the nasotemporal direction, and gray is the temporonasal direction, so the opposite direction, the same orientation, but the opposite direction. And you see that there's not much difference. We also tested other directions, and overall, there's no specific difference. So none of these were significantly different from each other when we look at the overall population response. So the population perturbation responses seem to be unrelated to the direction in which the animal was running. But then this is population responses. We also had the possibility of looking at individual neurons. And given that we know from decades of visual neuroscience that neurons can have an orientation or direction tuning, we also looked at the direction or orientation tuning of these neurons. So we asked two questions. So what is the at the individual neuron level, is there any dependence on direction? And is there any relationship to the direction tuning of the neuron? So I'm going to show a couple of extreme examples. First, this is a rare extreme example where the black dot here represents the orientation tuning or the direction tuning curve of the neuron. You can see that there's very little response of these neurons to the actual orientation or direction of the grating, but there's a very strong perturbation response in these neurons. But this was, as I said, a rare example, and this was not direction tuned. But we do have quite a few cells that have actually an orientation preference. This neuron is oriented in a 45-degree angle. And if you look at the perturbation response, it seems to actually follow a lot the orientation, the direction tuning curve of the neuron. So they seem to prefer the same orientation. So we then quantified this across the population of neurons we recorded. So on the x-axis here is the perturbation, the orientation where the perturbation response was maximal. And on the y-axis here is the preferred orientation of a neuron. And you can see there's a higher density here. It's better seen here where we looked at the difference between these two. And you can see that at around 0, where the perturbations match, the responses are more likely to be around closer to 0. This difference in orientation is more likely to be close to 0 compared to the shuffle distribution, which is in gray. I just want to add that I can't actually see the questions. So if, Saskia, if there are any relevant questions, can you please interrupt me?
Actually, given that pause, there was just a question that came up asking if you could explain the stimulus for this experiment again,
right.
If it's linked to movement, and how is the expectation being made through repeated stimuli, or if you just repeat that for people, that would be great.
Great, thanks for that. So there wasn't actually any link to the animal's movements. This was just an open loop experiment where we just played different directions in different, it's just like running a direction tuning curve. So it was not linked to the animal's movements. And we actually ran this, so I'm not showing these results, but we actually ran this in two conditions. One was where we had exposed the animal to the stimulus for over 10 days without the perturbations, and then we introduced the perturbations. And then we had another set of animals where we, on the first day, we started recording, and we started looking at the perturbation responses right from the first day. And actually, we've clubbed the data together because there weren't any differences between that difference in experience. But that's a really interesting, thanks for that question. Okay, So in general, what we found was that the responses, the perturbation responses tend to be larger in the preferred orientation of the neuron, which got us to question the fact that are there other visual response properties that match this perturbation response? And the most obvious one is actually temporal frequency, given that we are actually presenting a stimulus that's drifting for a constant period of time, and then we go to zero temporal frequency, and then we go back to a high value. So is there any tuning for temporal frequency? So I'm going to show two example neurons here. The top row here is a neuron which has a positive perturbation response. So that's this red curve here. And then this is an example of a neuron which has a slight dip in the response, which is the blue curve here. And when you look at the temporal frequency tuning curves, which are these curves on the right, you can see that this neuron actually tends to, seems to be more like a low pass neuron, which likes slower speeds, while this neuron actually has its peak around 3 Hertz, which was the stimulus that we were using for the characterization. And if you look at all the population of neurons, the red points here are these positively modulated neurons, and the blue points are the other neurons. And we can see that overall, the distribution seemed to be skewed towards lower temporal frequencies compared for the perturbation neurons as opposed to the other neurons, which are more uniformly distributed. So one other thing that we recently that was recently published, which was interesting, was that the sensory motor mismatch neurons seem to be more likely to occur in superficial layers compared to deeper layers. So we were really curious, like, okay, so would this correspond to the temporal frequency preferences? And actually, thanks to the Allen, we could actually go and look at the large data set and look at, is there any changes in distribution of temporal frequency with depth? And when we looked, so this was the imaging data set. And when we look at this, it was actually consistent with what we thought we might see, which was in lower depth in superficial layers, there seems to be a tendency to prefer slower speeds, as opposed to deeper layers, deeper depth of recording, where it was a much more uniform distribution of responses. So this was consistent with the fact that there are more I guess what we expect now, based on this, is that we should see more visual perturbation units in the upper layers as opposed to the deeper layers. So just to summarize this bit, we found purely visual perturbation can generate strong responses in V1, which are enhanced by running. They don't seem to depend on the running direction of the animal, and they're stronger in the preferred orientation of the neuron. And responsive units tend to prefer slower speeds in general for any other stimuli. So based on this, what we think is happening is that maybe sensory motor mismatch responses can be partly explained by just simple sensory responses that are enhanced by running modulation. And all the different properties that we might see could be, so the predictive coding aspect of this can still be achieved, but rather than having a complex model of having a difference calculated between the efference information and the sensory information, it could just be a simple distribution of visual response properties that add on the activation by running. So if you had about 30% of the neurons that like low temporal frequency, and even half of them are activated by running, that generates quite a strong, large population that has the properties that are required for this predictive coding of optic flow that has been reported. Okay, I'm gonna try and go through this last bit really quickly, as I see that I'm getting close to the end of my time. So this is actually just an anatomy study that we ran. Considering that we've been looking at a lot of modulation of responses in the primary visual cortex, especially to various different features such as navigation or movement and so on, where is all this information coming from? And while where V1 is projecting to is very well characterized, it's not as well characterized in terms of where the feedback is coming into V1 from. So we did this. So this was a study led by Mai Morimoto, who's a postdoc in my lab, and Emi Uchishiba, who was a master's student in the lab. And we did some very simple experiments where we injected retrograde traces specifically we injected chorotoxin subunit B or CTB into different regions of V1, and we looked at the expression in the cell bodies across different areas. So here's an example of some visual areas. You can see very nice retinotopy in the LGN. There's a couple of higher visual areas here. But we also have some non-visual areas like retrosplenial cortex, the anterior cingulate cortex, or even the medial entorhinal cortex. So things that get us excited about like maybe spatial information is coming from there. So, but to be able to quantify the responses across these, to quantify the distribution of inputs, we did a simple software pipeline where the first row here of the pipeline is just trying to calculate using simple image processing, trying to, from each brain slice that we did, trying to figure out where the centroids of the neurons are, the labeled neurons are. And then we used a variation of a program called SharpTrack to then register where each of these centroids are on the Allen CCF, or the common coordinate framework that you guys have established. So now that we have this detailed information, and the Allen CCF also comes with area coordinates, so we can actually identify where each cell body, each of these centroids that we identified, which area do they belong to. And when we do that, this is the kind of distribution that we get across all the areas of the brain. Unsurprisingly, there's a lot of the higher visual areas showing up, but we also have the retrosplenial cortex has a very strong projection back to V1. And there's also some non-visual sensory areas like auditory cortex and somatosensory cortex. And interestingly, there's some areas here around the entorhinal cortex and TEA, which is very close to the entorhinal, and the subicular complex, which are kind of interesting areas because they might be related to the spatial information present. Okay, so while this was just considering all the cells to be coming from V1 overall, we actually had a distribution of different injections. So if you look at the individual injection sites, this is how they look like. And this is the normalized azimuth retinotopy. Actually, this is data that also shared by Jack Waters at the Allen. So we could just take the coordinates and then map them to the retinotopic location for each of these injection sites. Now, based on this, we have, for every centroid that we detected for the cell bodies, we have their 3D coordinates, and we have the coordinates of the injection sites and the retinotopic position of these injection sites. So now we can try and look at, is there any correspondence? What is the distribution of these two looking like? So what we can, so when it comes to the source, what we can look at is like, if you had something that's very topographic, as what we're calling it, so that might suggest that an area in V1 is receiving projections from a very specific area of the source region, and similarly another region. On the other extreme, it might be that every region of the source area is projecting wherever in V1, so there's no specific topography, or it could be some kind of mixture of the two. So this is at the source end. At the target end, instead, we might have something like this, where one area of V1 receives a lot more connections than another area of V1. So there's a bias or a skew towards a certain area. And then we can actually complete this to look at all the different combinations of these possible. So I'm first going to talk about the source distributions. So looking at where the cell bodies are coming from. So this is just a reminder of how it looks like in terms of retinotopy, where the distribution of injection sites is. If you look at, this is just one example area, so we chose PM here. A gray point here represents that there is, that neurons from that region are actually projecting to all kinds of, so they're projecting across all the different injection sites. They're not skewed towards any specific retinotopic location in V1. A blue point would represent that it's actually skewed a little bit biased towards projecting to this region or the central visual field, and red represents that it's projecting to the periphery, peripheral V1. So if you look at all the visual areas, this is what it looks like. There is a slight correspondence to the retinotopy that we observe in the higher visual areas. But I think something that was really interesting was like, actually, if you look at any of these areas, almost any of these pixels that where we're averaging the information, all of them had neurons projecting to all the retinotopic locations. Occasionally, as are highlighted by these colors, we do see some biases towards specific regions, but overall they seem to have responses going, projections going across all the different areas. So this is the same thing in elevation. And this is just some higher visual areas. In general, you see it's mostly gray, but there is some little bias or a skew towards certain regions. So towards the center, towards the periphery and ventral in V1. In the interest of time, I'm actually going to skip the target aspect of it. I'm just going to go to this figure here where we basically look at the azimuth retinotopy of these injection sites and looked at their cell count and looked at the correlation. Is there a correlation between, so is there more projections to one specific area? And when we look at that, we find that some areas do have a significant correlation. So they tend to project more towards one side of V1 compared to the other. And that is also true in some of the higher visual areas. So, sorry, non-visual areas, so retrosplenial and ectorhinal and postrinal seem to have a little bit of a bias towards projecting to a specific region of V1. So overall, higher visual areas tend to be, so we almost never see this pure topography. Higher visual areas tend to be around here where there's a slight bias towards projecting to a specific region of V1, and there is some kind of semi-topography that's present. And in non-visual areas, they generally tend to be non-topographic, but some of them do have a significant bias towards projecting to a certain region of V1. So just to summarize this, we developed a pipeline to go from labeled cell bodies to the 3D Allen CCF coordinate framework. And we characterized the distribution of these projections from across the different brain regions into V1. And there's some semi-topographic arrangement of source cell bodies for higher visual areas. And many areas seem to show a correlation with the projection target in V1. I'm actually going to skip this bit and just go straight to the acknowledgement. So the study, so BonVision was developed, programmed and coded by Gonzalo Lopez, and it was run in collaboration with Sam Solomon's lab, and everyone in the lab was involved in testing and giving feedback on this, on BonVision. The parts of the vision and navigation was led by Mika Diamanti in collaboration with Matteo Carandini and Ken Harris. The part about the visual perturbation was led by Tommaso Mutsu, and finally Mai and Amy ran the last anatomy study. And I'd like to thank all the funders. And finally, given that it's Alan, I'd like to take this opportunity to thank you guys for all the data that you've shared with us, because especially through COVID lockdowns, a few of my students have basically exclusively been working on analysis of Allen data sets for some of their projects, and it's been really fun to work through that. And so thank you guys for all the data.
Excellent. Thank you, Aman, for that wonderful talk. There was a lot of great content in there, and it's exciting to see that you're using the data a lot. We have some questions. If people have more questions, add them to the Q&A box, but I'm going to start with the questions that are already in here, and we can see how far we get. The first question is from June, asking if does the V1 spatial modulation remap in different environments like hippocampus place cells do?
We haven't tried that yet. The one thing we've tried first is like we're trying to manipulate the landmarks and look at like how they follow the different landmark manipulations, but we haven't put them in a new environment yet. That is definitely an interesting thing that it's a good follow-up experiment that we should be running. It is actually planned. It's just been taken forever.
Sure. Our second question from Irina is active navigation that enhances spatial modulation different from just running? In other words, do you see enhancement of spatial modulation during replay when animals run, but movies that they see are not coupled with their running speed?
So I think that is exactly. So I only showed the case where the animals were running and we replayed it. So versus active navigation. So there is a greater spatial modulation when the animals were in closed loop and actually actively controlling the environment, as opposed to just running through the environment and experiencing the environment flow across its eyes. So that's a good question. So yeah, so this is indeed, you do need, so active navigation does enhance the spatial modulation aspect of the responses, yes.
And so do the animals run differently in active navigation? What's kind of is there kind of a difference there?
Right, so in general, so the difference would be that the speed of the animal and the speed of the virtual reality would not match each other. So there would be a slight difference. So I guess the running replay is a mixed condition where at times maybe the animal thinks that it's following, that the virtual reality is following its actions, and at times it might think that, wait, this is not doing what I'm expecting, I'm slowing down and this thing is speeding up. So I think it's a mixed condition when it comes to running, but the active navigation for sure is a pure condition where it is actually literally following what the actions that the animal is doing. So yeah, does that answer the question?
Yeah, I think so. I think, you know, there might be some subtleties in running, but I don't know that yeah, it answers it for me. If anyone needs more clarity, please add questions to the box. The last question we have here from Farzana, can you again show the slide where you suggested an alternative view over sensory motor mismatch responses, and explain again what your thoughts are? Are you suggesting some other mechanism instead of predictive coding for mismatch responses?
So I guess what I'm suggesting is, like, for predictive coding, you need to have cells that have certain properties, which is that when you have when you have a mismatch, you need to have a high activity. But what I'm saying, and if you're only going to have like 5 to 10% of the cells that do this activity, do you really need? So I guess the main hypothesis that we're trying to say might not be the case is that mismatch has been suggested to be an active subtractive process. So you look at the motor activity, you look at the visual activity, you are inhibited by visual activity and you're excited by motor activity. And that combination is what is giving you mismatch, is what is being proposed. What we're saying is that actually you don't need this kind of active suppression based on visual flow. You just need to have, you know, visual tuning properties where a static grating elicits a stronger response than a drifting grating. And this has been shown before. There's low pass neurons that we've known for multiple years. So it's, if you have a response like that and you combine that with some active modulation by running, you can get 5 to 10% of the cells that have the same properties that you would need for this predictive coding, without actually doing this active suppression by vision and activation by motor. So it's not that you can actually achieve the same thing with a much simpler mechanism, which is just the distribution of properties of the neurons, is what the idea is. I must add that it doesn't mean that there are no, so we haven't actually done the recordings in active closed loop conditions. So it's possible that there's additional neurons that are actually purely sensory motor driven, sensory motor mismatch driven. But what we're saying is that the neurons that we're finding, which are just visual perturbation neurons, they've under sensory motor mismatch conditions, they should be active because all the conditions that would activate them are active. So you're going to get quite a few of these cells showing up and maybe being mislabeled as sensory motor mismatch neurons.
Great. That was a very clear answer. Thanks. Those are all the questions. If anybody has any more questions, this is your chance to add them to the chat box. Oh, here we go. From Quanshin, have you looked at the laminar distribution of retrograde neurons in higher visual areas?
Yes, we have. It was actually something I was not sure whether I should show it or not, but anyway, we have most of the neurons from all cortical areas seem to be coming from deeper layers. So we've got about 80 to 90% of the neurons that we identified are actually deep layer neurons. And so in terms of, there's a theory about like which area would constitute as feedback or feed forward is determined by the ratio of how many neurons from the superficial layers are projecting there versus how many neurons from the deep layers. And it seems to be that most of these areas that we've been characterizing fall under the category of being feedback responses because we're getting these deep layer projections back into V1.
Excellent. Well, why don't we wrap up here? Thank you again for the excellent talk, really exciting work that you're doing, and really some very, very interesting ideas. A lot of things for me, at least, to think about. So thank you for joining us, and thank everyone for attending, and we will wrap up. Thank you.